Hello, my dear students. In this session, let us discuss about some of the historical aspects of microbial genetics, which led the foundation stone for the importance of microbiology in the field of genetics. The first in the list is Hershey and Chase experiment. And they conducted an experiment wherein they took T2 bacteriophage, which composed of DNA and simple protein, and they allowed this T2 bacteriophage to infect the bacteria. But before infecting, they conducted a small experiment wherein they took two batches. In batch number one, they cultured the bacteriophage in the media labeled with radioactive sulfur, incorporated with radioactive sulfur. In the batch two, they cultured the bacteriophage in the media with radioactive phosphorus. Let us see the reason why they had labeled it. Then they allowed it to infect the bacteria. The bacteriophage started to infect the bacteria and transferred its all the genetic content to this host cell. Then they centrifuged the media, the culture, they centrifuged. Then here you can see in this slide, you can see the batch one, which was sulfur labeled protein. After centrifugation, the precipitate, the biomass didn't glow, whereas the supernatant was glowing, which indicated that the sulfur radioactive labeled protein is in the supernatant but not in the culture of bacteria. Whereas the batch 2, the supernatant was not glowing, whereas the culture, the bacterial cell, they were glowing, which was labeled with phosphorus labeled DNA. And this observation led to a foundation stone so that the protein labeled bacteriophage couldn't transfer into the DNA, whereas this phosphorus labeled DNA was able to transfer the genetic material to the host cell. And here you can see the overview of this experiment. They cultured phages grown with radioactive sulfur and blended, followed by centrifugation, and radioactivity was not observed with the cell or a pellet, but it was observed in the supernatant. Whereas the batch 2 phages were grown with radioactive phosphorus, and here they cultured this phage, phages into this media and they centrifuge, and they observed that there was no glow or fluorescence in the supernatant, whereas the fluorescence was observed in the pellet. So, which gave or indicated that the genetic material is transferred to the host genetic material only with radioactive sulfur. So, this led them to conclude that DNA was the genetic material but not the protein, which was really essential during those days. And the next experiment was Griffith experiment. The Griffith experiment also is one of the most important experiment in the field of genetics and microbial genetics. Griffith conducted an experiment, and during the experiment he wanted to develop a vaccine against pneumonia disease. Hence what he did, he took two strains of Streptococcus pneumoniae. Strain S he injected to the healthy mice, and he could see that strain S was very lethal and killed the mice. Whereas the living R strain was injected to the healthy mice, and there were no death or no infection caused. And when the S cell, which was very lethal, was heat killed and inoculated to the injected to the healthy mice, then no colonies were observed, or no, the mice didn't die, but it was healthy enough. When they mixed heat killed S strain along with R strain, they could see that mice was infected with pneumonia, and this S strain and R strain were isolated from the dead animal. So this experiment gave an idea that there is a transforming principle which is taking place, which is making this to happen. So let us see the overview. What is R strain? R strain, when grown in a petri plate, the R bacteria formed colonies or clumps of related bacteria that had well defined edges and rough appearance. Hence, it was labeled as R strain. The R bacteria were non virulent, meaning that they did not cause any sickness when injected to the mice. Whereas the S strain, here it was rounded and smooth, the colony was smooth, and hence it was abbreviated as S strain. Whereas it was virulent. The same here you can see living S cell injected to the healthy mice, mice died due to its virulence. The living R cell injected to the healthy mice, it didn't die. When the heat killed virulent strain of S was injected to the healthy mice, the health, the heat killed S strain was injected to the healthy mice, then there was no infection, even though the S strain was a virulent. But after the treatment of heat, there was no infection. Whereas when heat kill strain along with R strain, which was not showing any infection, was mixed and injected to the healthy mice, it showed the infection and mice died. So they came to a conclusion that they concluded that R strain bacteria must have taken up what they called as a transforming principle from the heat killed S bacteria, which allowed them to transform into smooth coated bacteria, which was the virulent factor. So this was one of the experiment which was really essential in order to give the confirmation that there is a transforming effect. And the next important discovery was DNA structure. The DNA structure, as we have already studied in our PUC and all, it is a double-stranded DNA, double helix, and it contains each strand contains a ladder which is made up of phosphate group and deoxyribose sugar. And the steps of this ladder is made up of nitrogen bases ATGC, adenine, thymine, guanine, and cytosine. And the pairing of this nitrogen bases is with the help of hydrogen bond. So this is an overview, and this gave a complete picture of how DNA looks and what are the characteristics, what are the components of this DNA, what is the structure of this DNA, which was very essential in order to carry out various scientific discoveries. So during those days, these three experiments played a very major role in the field of microbial genetics as well as in the genetics. Here you can see the overview of this chemical structure of DNA, and along with this, let us study some of the important genetic tools which are essential in those days as well as in the modern science. PCR, polymerase chain reaction. The purpose of this technique is to amplify a specific DNA sequence. It is one of the most common instrument found in research laboratory. The application of this instrument is, here you can see DNA cloning, to study gene expression, analysis, genotyping, DNA sequencing, etc. It is one of the most fundamental and basic instrument to be found in research laboratory, especially in molecular biology labs. The next come in the list is, before moving to the next segment of the instrument, let us see how this PCR works. Here you can see the PCR. Here you have a template DNA. To this template DNA, you will add the specific primers and DNTPs, which are nothing but the nitrogen bases. Along with this, you add Taq polymerase and mixed in a buffer in a PCR tube, and this PCR tube is placed into this PCR instrument, and PCR is carried out. During the PCR procedure, there are different thermal cycles are followed. Here you can see there are three basic steps of PCR. One is denaturation. What happens? First, there is a rise in temperature which results in the separation of DNA fragment, and then these primers goes and attach to the uh, sub, specific complementary uh, base pairs, and there is again annealing factor, and which results in again heating, which results in extension of this DNA from 5 dash to 3 dash and from 3 dash to 5 dash. So like this it starts growing, and we get the multiple copies of DNA with the help of PCR. The next important invention was plasmid vector. Plasmid vectors, as we have already discussed what are plasmids, and plasmid vectors plays a very important role in the genetic engineering. Here you can see the foreign DNA is integrated into the plasmid, and this plasmid is inserted to the bacterial cell, and when the bacteria starts multiplying, the number of copies or clones of this specific foreign DNA is produced. So this plays a very important role in genetic engineering. Restriction enzyme is one of the important enzyme in the field of biology. They cleave the invading bacteriophage DNA in order to prevent the infection of the bacterial. This is one of the application of this. And CRISPR, this is one of the important tool wherein they try to correct the mismatch or unwanted sequence in the DNA. Here you can see there is unwanted sequence in the DNA template. What they do, this CRISPR, it has a two there are two components in this. One is Cas9 enzyme and second is guide RNA. What they do, they go and try to find the mismatch or unwanted sequence, and then they try to cleave this. After cleaving, they try to add the desired DNA sequence so that a desired gene can be expressed and desired protein can be obtained. Next in the list is DNA sequencing. DNA sequencing is an instrument which is used to analyze the percentage of nucleotide present, so that based on this percentage, a specific genome or a DNA can be matched with the given databases and can identify. For example, if you are working on an unknown bacteria or unknown fungi, you will try to isolate the DNA, try to do PCR, then you go for sequencing. During the sequencing, you will get the complete information of the nucleotides, and based on this percentage, you can easily identify using NCBI tool. RNA interference. This is one of the important uh, component in molecular biology. Here, it chops off or knockdowns or silences a particular gene expression. So this is really handy when it comes to in the case of deleting a gene segment which is capable of causing infection or which is capable of causing disease. So this was one of the greatest discovery which has led to map the healthy genes and compare them with the disease causing genes. Genomic library and cDNA library. The name itself indicates library. They try to store the DNA in the form of library so that anybody who wish to work on a specific functions of the DNA, they can pick up that specific DNA and they can start working on it. So how they do, like genomic library, here you have a cell, the DNA is extracted, restriction and enzyme cuts this DNA, and the ligation process takes place and it is transferred into uh, the bacterial genome or a plasmid and insert it into the bacteria, and these bacteria are preserved in the form of library. And here along, it is a complete genomic sequence. This is how the genomic library is used. Whereas cDNA library, here from mRNA sequence, they try to generate complementary DNA, and the rest process is same. They use the vector, the ligation, they insert this specific uh, sequence of complementary DNA to this vector, and from there ligation takes place, and this plasmid is uh, in, inoculated or inserted into the bacteria, and the bacteria are preserved in the form of library. Whenever if anybody is looking to work on a specific expression of the gene, they can easily take that bacteria and they start uh, working on that genetic information. DNA microarray, it is it is also one of the technique in the molecular biology where you can easily identify the cancerous or disease causing cell and normal cell. You can easily distinguish with the isolation of mRNA and reverse transcriptase enzyme, which is labeled, and this creates a complementary DNA, and this complementary DNA probe are attached onto the wells of this slide, and whenever you add your sample, this targeted probes goes and binds to the complementary sequence, and it glows or it emits light. Based on that, you can easily identify or distinguish cancerous cell and normal cell in a tissue. So quantitative PCR is similar to the in the case it's similar to conventional PCR, but the only difference here is you can easily monitor the live reaction of the PCR and how the aligning is taking place and how it is multiplying, and you can easily quantify the sequencing, or you can easily quantify the amplification process. DNA fingerprinting, it is very common nowadays. You take any sample, you you cut the sample with the help of restriction enzyme, and you carry out electrophoresis PCR, and you place that fragments to a membrane or a chip. Then you add your identifying sample. If there is if there is a binding, then the radioactive labeled probe will glow, and you can match this with uh, the given database, which help us to identify, especially in the case of crimes. If you get any samples, biologically biological samples such as hair, blood, any sample, you can easily extract the DNA, and you can easily amplify the DNA, and you have a complementary membrane with the DNA probe. If you add on to it, then you can easily see the amplification of this the target DNA. So these are some of the important tools in the field of molecular biology and microbial genetics. Before we could start this microbial genetics, we need to have an overview of this. So today's class was an important session in order to give you a superficial and overview of the history and development of the microbial genetics and the genetic tools used. So in our future classes, let's see other important aspects as per your syllabus. Thank you.